Hello students, welcome to DAV Sri Vishnu Vidyaniketan online classes. We are discussing a series of experimental studies. We have go through so many biological demonstrations. So today also I am ready with awesome experiment. In this module you could able to learn about bloodstream. Let us see how to check the blood groups. Blood group identification activity. Before going to the video, I want to tell you about the scientist who contributed about blood groups. He is called Landsteiner. He identified blood groups for first time. He identified peoples containing A, B, AB and O groups respectively. Different persons containing different kinds of blood groups with different RH factors of positive and negative. The blood groups are vary as A positive and negative, B positive and negative, AB positive and negative, and O positive and negative. Let us see how to recognize the blood groups. The material required for identification of blood groups, it is very simple. We need blood group identification kit, glass slide, alcohol or spirit, needles and cotton. Blood group identification kit, which containing 3 small bottles. Those are blue, yellow and transparent in colors. So, these are respectively anti-A, anti-B and anti-D serums. Anti-A serum is responsible for the grouping of group A, which is blue in color. Let's see the identification of blood group A. Blood agglutination in anti-A, which we serum taking blue in color, just put a drop of blood into that. If the serum agglutinates the blood sample, then it identified as group A. Let us see the reciprocal one. If we take another serum or another blood group which not able to agglutinate that. Let's see one example for that. Take serum A and take another kind of blood group which is not group A, which is not agglutinating here. So it is not blood group A. Anti-B. It is a serum which is possessing yellow in color, which is responsible to recognize the group B blood. So take one drop of serum B and put one drop of blood into that. If the serum agglutinate the blood drop, then we good to know that it is blood group B. If it is not agglutinating the blood drop, so we have to identify that it is not at all a blood drop which belongs to blood group B. So, like this, we have to recognize the blood groups A and B. So, we have to take 2 drops anti-A and anti-B. If anti-A get agglutinated, then it is recognized to be blood group A. If it agglutinates anti-B, then it is recognized as blood group B. If the blood sample agglutinates both A and B, then the blood group is recognized to be blood group A and B. If the sample which doesn't agglutinate neither A or not B, then it is recognized as blood group O. So, let us take some more examples. These are different slides taken from different blood groups: Group A, Group B, Group AB and Group O. Let us check it out. The RH factor. The serum possessed in transparent color, which is anti-D, and it is responsible for RH factor, which is giving a positive and negative to respective groups. So, if we have to know the blood is positive or negative, take one drop and put one drop of blood into that serum. If it agglutinates, then it go with the positive. It is a slow process and it will take some time to give the result. We have to wait for some time. Here the blood sample which taken, it is agglutinating. So the given sample RH factor is positive. Suppose if you are taking the person's blood from group A, then it is getting agglutinated in this serum D, then it is known as positive one. If it doesn't agglutinate in that serum, then the blood group is about RH factor negative. Means the blood group doesn't containing any RH factors. I hope you understand about blood groupings with RH factors. Now let's identify the blood group for one person. For this we have to take one slide and take 3 drops respectively from the 3 bottles, anti-A, anti-B and anti-D. Select one finger and sterilize the finger using one cotton pad. We have to use alcohol or clinical spirit for that. Let it dry. Take one surgical needle and just pluck it out. Take bloodstream and inoculate blood drops in respective drops of anti-D, anti-B and anti-A. Mix the bloodstream with the anti-A. If it agglutinates, then it is confirmed that the sample blood is group A. Then mix the bloodstream with another drop which is anti-B. If this drop is also agglutinate, then we can confirm the bloodstream sample is about AB. If the sample is agglutinated in anti-D also, then we could recognize that AB positive. But here the sample taken which is A positive. So this is very interesting experiment, isn't it, children? That's all for today. I will meet you with another new topic in another video. Until then, keep learning, keep getting knowledge. Thank you, children.